Good afternoon, everyone. I'm Terren Niethamer, a postdoc in Ed Morrissey's lab, and today I'll be telling you a little bit about our work studying pulmonary endothelial cells. In the Morrissey lab, we're fundamentally interested in lung regeneration, and the lung is not continuously regenerating itself at homeostasis. It's pretty quiescent, but if it's injured, it contains facultative progenitor cells that can be activated to repair it. And we're preliminarily focused on the gas exchange unit of the distal lung, the alveolus. And the important cells for gas exchange are the alveolar type 1 epithelial cells and the pulmonary endothelial cells. And the alveolar type 2 epithelial cells can actually serve as these facultative progenitors to regenerate the epithelium. Zooming in on this region, although capillary endothelial cells are one half of the gas exchange machinery in the alveolus, we actually don't know a lot about them. And we don't know if there are specialized cells that participate in gas exchange or whether there are specialized cells that regenerate the endothelium after injury, or perhaps signal to epithelial cells to restore this key interface. And so to answer this important question, of course, in today's world, we turn to single-cell RNA sequencing. As our model, we're using the mouse lung, which is very similar to the human lung in its cellular composition. And to injure the mice and look at regeneration, we give them the flu and follow them out to 14 days, a point at which regeneration is actively occurring. And when we do this, we isolate the mouse lungs, make a single cell suspension, and isolate CD45 negative non-immune cells for single cell RNA sequencing. And looking at the difference between the control and the injured lung, you can already tell that the flu by H&E is a very heterogeneous injury. There are these densely injured regions and some regions that look less injured. And we actually like this as a model because it helps us to know that some of the same cell types will still be present. The lung is not totally destroyed, but we might also find some new cell types. And looking at the control and the flu-injured lung, our first result, which we were very excited about, is that we do see endothelial cell heterogeneity. And I've highlighted the endothelial cells here in color. And notably, we see many of the same populations under both conditions. So what are these cells? Of course, all endothelial cells express the general marker PCAM1 or CD31, but we see macrovascular endothelial cells, both venous cells marked by expression of VEGF-C and arterial cells marked by expression of BMX or CXCL12. And when we look in actual mouse lungs using RNA-scope, for these markers, we can find these cells in large vessels and not in the alveolar capillary space. We also find microvascular endothelial cells, which in addition to PCAM1, most of them express GPIHBP1, and a few also express IFI47. And again, when we look in the mouse lung using RNA-scope, here for GPIHBP1 and both PLVAP and PCAM1 as general endothelial markers, we find GPIHBP1 expressing cells in the alveolar space, but not in large vessels, as we would expect for microvascular endothelium. But perhaps you've noticed, and we noticed too, that there was a fifth population of endothelial cells here. And these cells express high levels of CD34 and the carbonic anhydrase CAR4, as well as high levels of signaling molecules KDR or VEGFR2 and endothelin receptor B. We wondered what these cells could be and whether they had a distinct functional relevance in the lung. But first, I always have a healthy dose of skepticism about what I'm seeing in these single cell gene expression plots, and I wanted to know if this is really a distinct cell population. So I started by just looking for these cells using RNA-scope. And looking at cells that co-express EDNRB and CAR4, I found these cells scattered throughout the distal lung, but never in large vessels. And in data I'm not showing you, I'm actually able to isolate these cells using FACS with the CD34 antibody and show that they do express higher levels of these genes that I find in my single cell RNA-seq by QRT-PCR. But what are these cells actually doing? And to answer this question, I turned back to our influenza injury model to see what they do after the lung is injured. I perform the same flu injury of the mice and isolate control and injured lungs at 14 days post-injury. And in the normal lung, IHC for CD34, just as sort of a first look, shows that these cells are distributed throughout the alveolar space, as I see with the CAR4 RNA-scope. However, in the injured lung, when I look at these regions of severe alveolar damage, which are here outlined in a yellow dotted line, I see that adjacent to these severely damaged regions, there's an increase in CD34. And this could point to several possibilities. These cells might migrate to regions of dense injury. They could upregulate CD34, of course, or CD34 expressing cells might proliferate at these injured regions. And I decided to look more at proliferation, and here's why. Going back to our flu single cell RNA sequencing data, we actually found a population of endothelial cells that we labeled proliferating endothelial cells. And that's because they express high levels of genes associated with proliferation, topoisomerases, this regulator of cytokinesis, Ki67, but they also express endothelial cell markers. And so I wondered how endothelial cell proliferation might contribute to injury. And at this point, I actually hypothesized that perhaps these CD34 high cells are serving as an endothelial progenitor. So I returned to my flu model, but this time I gave the mice EDU in their drinking water from days 7 to 14 to assess proliferation during this time period. And when I isolate CD45 negative cells from a flu-injured mouse, and then separate the CD31 expressing endothelial cells into CD34 high and CD34 low cells, and then look at EDU incorporation in each population, what I saw surprised me. I actually saw that both CD34 high and CD34 low endothelial cells were incorporating EDU and therefore were proliferating during this time period. And when I compare this to the control, I see that not only does endothelial proliferation as a whole increase in these mice, there is an increased number of CD34 high endothelial cells, confirming what I saw in my IHC data and quantifying it, which is great. But also there's an increase in both proliferation in CD34 high cells and CD34 low cells. And this was very exciting to us because it indicated that there's not one endothelial cell subpopulation that preferentially proliferates after injury, but rather multiple populations can perform this role. So I've shown you that endothelial cells in the lung are heterogeneous, and that these CAR4 high cells are a specialized population in the distal lung. We know that they localize through the alveolar space at homeostasis. We know that they express high levels of signaling receptors, such as VEGFR2 and EDNRB, and we know that they localize to these regions of dense injury after flu infection. But we don't actually know what they're doing. And so I just want to say really quickly that I've developed a CAR4 CRE-ERT2 and an EDNRB CRE-ERT2 mouse. I'm currently still characterizing these mice, but I'm hoping to use them along with some other mice that can specifically target other EC populations to find out what these cells are doing after injury. And while we learned that endothelial cell proliferation increases dramatically after injury, it's not one subtype that preferentially proliferates. So of course, some remaining questions. I want to know what the relationships are between these differing endothelial cell subtypes, the functional significance of the receptor expression in the CAR4 high cells, which I'm looking into now. And of course, I would like to know whether these cells are signaling to other alveolar cell types, such as the epithelium, and what role this might play in regeneration. So thank you all very much for your attention. I appreciate it.